Recently, an unusual paper came out showing that vaccinated people, after a certain number of boosting, can have this unusual antibody class switch. So, what is that all about? This is what we're going to be talking about today. My name is Dr. Mikola Rashek, in a slightly different spot than usual. Nevertheless, still doing lots of hiking, lots of walking, and let's get going. So, this is the first paper, as far as we understand, that actually determined to look at longitudinal effect of vaccination on what might be happening with the antibodies in vaccinated individuals. And what this paper showed that basically there is a class switch. So what happens is there's different number of types of antibodies. So you might have heard before discussing IgMs and IgAs, and the most common ones and the ones that we're most familiar with are the IgGs. Now the IgGs themselves, there's still 4 different types as well. Now, typically what happens is you start with a specific class. Normally the first one is IG3. And the reason why is because the gene that is responsible for production of these antibodies, and this class specifically refers to the stalk of the antibody. So you know how the antibody has that Y shape? The stalk, the bottom half of that Y would be the stalk. And this is where the class which takes place. And we're going to be talking about that because that's actually important. So the gene that is responsible for coding these, it has number of these genetic elements coding for these stalks. Those and they're referred to as gamma 1, gamma 2, 3, and 4. And the way they are organized is that the gamma 3 is first. This is then followed by gamma 1, gamma 2, and gamma 4. Now, the B cells, which are normally responsible for producing antibodies, they can switch from the first one. Normally, they employ the first one, which is gamma 3. Over time, they can switch to the other classes. And that switch is dependent on it can be activated by these mutating enzymes called cytidine deaminases, sorry. And basically these are enzymes that can mutate the genetic material. And this is how we can get this variety of antibodies, because you can produce B cells that will have memory to make sure that they produce specific antibodies against viral proteins that were attacking us. But these antibodies can be slightly mutated by these enzymes present inside those cells to increase the variety of antibodies, as well as to be able to respond to changing variants. These cytidine deaminases also are employed by inflammation, and this is one of the ways we also fight pathogens as well, by mutating their genetic material. And this is also one of the reasons why inflammation in general can be dangerous, because you don't want to have too much of it, because the mutation can also happen inside your cells as well. So this is one of many different reasons why inflammation in general is dangerous. And if you can help it, you want to avoid inflammation as much as possible. But when it's needed to help you fight something like a dangerous infection, this is where it can be useful. Okay, so let's go back to this. What happens is that these mutagenic enzymes can lead to eventual switch from antibody class one class to the other. This is referred to as class switch recombination. And this is what was observed in this study with approximately 30 healthcare workers who were vaccinated. And what was shown is that at the beginning of the vaccination, in the study, what the authors did, they collected blood samples 10 days post every vaccination that these healthcare workers did, which was up to 3 different vaccinations, 3 different shots. So 1, 2, and then a booster, so 3 different shots. And then they also collected blood samples approximately around 200 days after each vaccination as well. The vaccinations regimen was between 3 to 4 weeks between the first shot and the second shot, and between 5 to 7 months after between the second shot and third shot. So that's kind of the history that was employed here. Now, what was observed that at first, when after the first shot, basically there was no IgG antibodies, and that's normal. This is basically the predominant antibodies were Ig1 and 3. There was a little bit of IgG2 class, and basically no IgG4 class, and that's normal. But what was observed that predominantly, especially after some vaccines after the second shot started to demonstrate switch towards the IgG4 class, and this specifically increased a lot more after the third shot. So we're going to be talking about what does that possibly could mean. Now, what's interesting though, that if the vaccinees were also infected, so if they had a breakthrough infection and they were infected by the natural virus, then that compounded the effect and it enhanced the class switch towards IgG4. Okay, so that was interesting because it showed that in just natural infection was compounding this, and this is fairly unusual because we'll get into this right away, because normally this class switch is not seen. That's not a typical thing, not even after natural infection or after multiple vaccinations. This normally does not occur. So what does this mean? Just checking the mic is still on. What does this mean? Clearly something different than normal is happening. They did not test the unvaccinated individuals. It would be interesting to see whether this has anything to do with the type of virus, or whether this is specific to the vaccinees. But we don't know. But if we had to guess, typically this is not expected after this type of class switch to take place after infection. Now, what they did is they were looking at Pfizer mRNA vaccinated individuals. So they did not look at the mRNA from Moderna. And the authors even mentioned it would be interesting to see if this is also potentially can be observed also with the Moderna vaccines as well. And the reason why is because there could be spike protein differences as well between these two vaccines. So is this, but they do think this is specifically mRNA vaccine specific, because they also looked at some participants who had only one shot of the mRNA vaccine and then one shot of non-mRNA vaccine, and they didn't show this type of behavior. There was no such a rapid or no such a large increase in the switch from the IgG3 and 1 towards IgG2 and 4 class of antibodies. They studied this in another cohort. So the first cohort was about 30 individuals. They studied it in one more cohort of about 40 individuals. And same kind of behavior was observed. With the second cohort, they actually provided us with a quantification number in terms of how much of a change is, and they mentioned there was about 40-fold increase in those IgG4 antibodies post third shot. Okay, so then here we go. Now, what does it mean? Well, the authors did not comment on that. They don't know whether this affects the capacity of the disease, but they did mention that all of the individuals they tested, the fact that there was this increase in the IgG4 antibodies, it did not seem to result in a more increased disease. Why do I mention this? Is because IgG4 antibodies, they're actually still somewhat mysterious. We are only starting to learn more about this. So we still have to know exactly what they do, how they function, etc. And there, as a consequence, there's not too many studies afforded to us to know what is going on. But there is earlier studies, one that came from Brazil, for example, that showed that increased level of IgG4 antibodies in COVID-19 patients correlated with more severe outcomes, okay? And another study showed that the higher the ratio between IgG4 to IgG1 antibody, again, the worse outcomes you get to see. But the authors of this particular study, they did not observe that in the results of in their own results with their own patients that they were analyzing. So, what do we know about these IgG4 antibodies? Well, typically, what they do is they increase tolerance and towards an antigen. So, and as a consequence, what you do when you use it, when you use these antibodies, IgG4 antibodies, is when you want to clear something out of the system, but without necessarily causing the inflammation associated with clearing a pathogen. So a good example of when you see this type of tolerance is beekeepers when they actually want to clear the antigen. Well, that would be like the bee venom, but without having the constant activation of the inflammatory pathways. Because what happens normally, like, why do we need antibodies? Obviously, they will bind to the pathogen, and let's say if it's a virus, it will help to block the virus from being able to then subsequently infect individuals, right? But then there's other things that happen to it. It's not just that you want the antibodies to bind the virus. Then what you can do is you can clear the virus that is coded by antibodies. So not only do you want these antibodies to prevent the virus from infecting cells, that's obviously neutralization, but then you also want to get rid of this virus. And there's multiple immune events that happen um, that help to clear it. So one of them is phagocytosis. That's mediated by specific immune cells. So what they do is they recognize the stalk that of the antibody. So remember, if the antibody has a Y shape, the tips of the Y will bind, say, something like the spike protein, but it's the stalk that is then recognized by the immune cells, and they can then incorporate that and then destroy it. So then this is referred to as antibody dependent phagocytosis. Then there's also antibody dependent cellular uh, um, cytotoxicity. So basically, that's where you destroy the entire complex. And then finally, antibody-dependent complement deposition. And complement are specific proteins that can accumulate and lead to also target the virus for the destruction as well. But these events are inflammatory, so they provoke inflammatory response, and they can also be destructive to the tissue. So this is so what so normally you want to have this happen to help to clear the virus, right? To help to clear it. And this is why you can have switch to something like IgG4 class, because the stalks of the IgG4 antibodies, they are not recognized by immune cells the same way that the stocks of other classes of antibodies are recognized. So, as a consequence, these immune cells have receptors. They're called Fc gamma receptors. They recognize specific component of that stock. The stock is called Fc. It stands for F crystallizable. That's what that stands for. And IgG4 antibodies, they no longer typically are recognized by those receptors on the immune cells. So that means if you are switching to these antibodies and whatever the binding will not be recognized by cells and will not be cleared by the cells. So what does that mean? We don't know what that means in the context of the spike protein, but normally what we are learning with the IgG4 antibodies is that it has emerged that they also are involved in a number of diseases. So they are involved in autoimmune diseases as well as what is referred to as related, basically IgG4 related diseases. And with the IgG4 related diseases, this is where you have very high increases in these antibodies in the circulation, and what happens, in such diseases, there's a multi-organ tissue damage, and these tissues are infiltrated by white blood cells, as well as those B cells that produce those type of antibodies. But we don't know exactly what kind of antigens these antibodies are recognizing. They don't seem to be recognizing very specific antigens. Now, with the autoimmune diseases, you don't see these spikes or increases in IgG4 antibodies, but they definitely seem to be targeting specific antigens. And what happens is they prevent other antibodies from doing their proper function. So basically, IgG4 antibodies can compete. They can compete for binding and prevent other antibodies from doing their job, and that in itself can result in autoimmune disease. So this is we don't know yet what it means to have this switch towards IgG4 antibodies in vaccinated individuals, but it could be problematic if it leads to tolerance. You might not want to have a tolerance towards something like spike protein, and the reason why is because there's clearly slowly emerging evidence that spike protein could be very damaging to the circulation. Okay, so then I'll be talking about that in the future videos as well. So tolerance towards spike protein might not necessarily be a good thing. And the authors were able to show that this increase towards the IgG4 antibodies is definitely mediated by also an increase in memory B cells inside those vaccinated individuals that specifically produce those IgG4 antibodies. They were able to demonstrate that, and they've confirmed it with sequencing, and they also confirmed that as a consequence, these IgG4 antibodies no longer can, in their test, they were no longer being uptaken by cells that are normally would uptake antibody complexes for phagocytosis or complement deposition. They did really interesting experiments where they took microbeads that were coated with spike protein, and these were fluorescent, and they were able to actually show that when you coat these with those spike proteins with antibodies, um, with the IgG3 and IgG1, there was still everything was happening okay. And so then the cells were able to phagocytose these antibody and spike protein complexes. But when they used IgG2 or IgG3, those antibody dependent cellular events were decreased. So clearly that means is that if we have this switch towards IgG4 antibodies, it will result in reduced clearance of the pathogen inside our body. So that's what that means. Obviously, tolerance. So, but these are important events. These antibody-dependent effector cellular events are, as they are called, they are important for clearing the disease, right? So what the authors suggest that perhaps there's still these IgG4 antibodies still could be neutralizing the antibody, and indeed all of the vaccinated individuals they studied after the third booster, the neutralization still increased of their antibodies towards the virus, but the cellular events that clear the antibody virus complex will reduce. So is that enough? Time will tell. We'll need more studies. As I mentioned, this is the first of its kind. And this might be the reason why this has never been seen before, where you actually study this for a long period of time as to what's happening with the antibody class switch. And the reason why is because this might not have been expected at all. Because as I mentioned, that's not a typical event after either infection or vaccination. So time will tell what it means. But we need to be vigilant on this because we could be potentially seeing an increase towards events that are either tolerogenic or autoimmune, and that might not be a good thing with regards to something as dangerous as spike protein. So, okay, that's all I have for you today on this topic. Thanks once again for everyone for supporting the channel, and thank you everyone for sharing the videos. Thank you for leaving your questions. We still have the ongoing COVID Q&As, so I look forward to seeing some of you there. And if you want free tickets to that, Please subscribe to our newsletter, and everything is in the description below. And I look forward to seeing you next time. Bye, everyone.